Students, this class is for fourth semester students. Lecture number 4, continuation of plant tissue culture. In which we have to discuss about edible vaccine in this chapter. Before that, I would like to have a discussion in the questions repeatedly asked in this chapter since 2019. They have asked 5-mark question, 10-mark question, even in a question they have asked 3 questions also, some questions, some year they are asking. So it's an important for the students to study this 4 lecture in detail. Nutritional requirement of plant tissue culture for 5 mark in February 2024. Type of tissue culture 5 mark in the same question paper. Tissue culture as source of medicinal compound 2-mark question. It is also in the same question paper they have asked. So you see more than 12-mark questions, 12-mark questions they have asked in a question paper. Now then cryopreservation they have asked 2-mark question in February 2024. And in 5-mark in the question they have asked, this is about 10-mark question in May 2024. Write a short note on edible vaccine. Write a short note on application of plant tissue culture. Define plant tissue culture and explain in detail about types of cultures, nutritional requirement for growth and their maintenance. In March 2023, 10-mark question they have asked. Application of plant tissue culture in pharmacognosy they have asked in July 2023 in 5-mark question. Phases of growth in plant tissue culture July 2023 for 2 marks they have asked. This part we have not discussed. I have added in today's lecture actually. Define edible vaccine. Today we have to discuss about that in November 2022 for 2-mark question. Explain the various phases of growth in plant tissue culture in June 2022 for 5-mark question. So I have added this portion in today's lecture actually. Discuss the various nutritional requirement in plant tissue culture December 2021 for 5-mark question. What is explant? The definition for 2-mark question December 2021. Write the application of plant tissue culture in pharmacognosy April 2021 for 5-mark question. What is plant tissue culture? Mention the types of cultures. Write a note on historical development and application of pharmacognosy. July 2019, 10-mark question they have asked. What are explants? In July 2019, 2-mark question. Write application of plant tissue culture in pharmacognosy. December 2019, 5-mark question. Define and classify plant tissue culture December 2019, 2-mark question. So I have seen that most of the time they have asked a minimum a question or 2 or 3 questions have been asked from our chapter. Now we'll go to the vaccine, especially we want to take the edible vaccine. So we should know what is a vaccine and then edible vaccine. Vaccine, the points should be remembered as a definition. A biological preparation improves immunity to a particular disease, resembles a disease-causing microorganism, weakened or killed form of microbe, stimulate the body's immune system to recognize the agent or the foreign body, destroy it and keep a record of it for later encounters. Regeneration will take place. Reduced mortality rate. Control various infectious diseases. These are some of the points which you should mention when you want to write about vaccine. You can write as a sentence each and every point which I have mentioned here. So, a protein which acts as a vaccine with all the points above, present in food and consumed as an internal composition of a food, is known as edible vaccine. So, you have to write what is edible vaccine means. Simply if you write it is edible vaccine which can be consumed means will not be sufficient. You should write what all the points needed for vaccine which will be present in food and consumed as an internal composition of a food is known as edible vaccine. That's the real picture of one vaccine, edible vaccine definition. So edible vaccine, you see, I have taken a picture to give a simple idea and to write it in the form, and to, if there is a 10 mark question or 5 mark question, a student should be able to write. See, first I have made a round here in the picture stating that taking a plant infected bacteria in a plasmid as a transfer vector, human infected virus and a gene of interest together, there will be a modified plasmid can be prepared in the in vitro study of in a laboratory tissue culture lab, and then transfer to modified plasmid into a plant cell. That you have to select a plant and the transfer of modified plasmid, already modified plasmid into the plant cell, and plant transformation with desired gene, and then the callus culture is developed. Can you see that? The callus culture, cell division takes place. And it can be after cell suspension culture, a large scale of production of edible vaccine plant can be developed. From that we can feed animals orally and feed human orally. So this is what the detail of a procedure, a outline of a simple procedure. What do you mean by an edible vaccine and how it can be prepared in tissue culture, and what is its relationship with the tissue culture in vitro study. Edible vaccine, why you need an edible vaccine? Immunization through DNA vaccines is an alternative, but it is an expensive approach. Edible vaccine gives cost-effective, easy to administer, easy to store, and socio-culturally readily acceptable vaccines for their delivery systems. Oral vaccine provide mucosal immunity at various sites of selective antibodies. Do not or don't need to worry about reuse, misuse, and lack of sterilization, thus low risk of infection. So these are the points which you should remember. So first one is edible vaccine. Why? DNA vaccine, expensive approach, cost effective, easy to administer, easy to store, mucosal immunity, socio-culturally readily acceptable, don't need to worry about reuse, misuse and lack of sterilization, low risk of infection. You should remember these points. An ideal edible vaccine should have the following properties. Safe. The vaccine should not be toxic or pathogenic and should have low side effect. It should not cause problems for people with compromised immune systems. Effective. Ideal edible vaccine should be effective. The vaccine should produce long-lasting immunity. An ideal edible vaccine should be easy to take. The vaccine should be easy to consume. Ideal edible vaccine easy to store. The vaccine should have a long shelf life and easy to be stored. And an ideal edible vaccine should be affordable. The vaccine should be less expensive. An ideal edible vaccine should be socially justifiable. Edible vaccine are especially promising in developing countries. So these all are the points which you should specify very clearly for an ideal edible vaccine. Vaccine agents can be delivered orally by administration of transgenic edible part which are developed using the methods of molecular biology. The genes of interest can be introduced into the plant where they are expressed in the plant tissue, including the edible components. This process is known as transformation, and the genetically modified plants are referred to as transgenic plants. These genes encode protective vaccine antigens from viral, bacteria and parasitic pathogens that cause disease in human and animals. The vaccine can be delivered by intake of the edible part of the genetically modified plant or the high yield production of refined protein for oral delivery. Edible vaccines are like unit preparation as they are built to contain antigens. However, bear no agents that might change whole pathogens to cause harmful effect to humans. Methods of transgenic or transformation of DNA gene into plants. You have to make transformation of the gene or the DNA to the plants. Then only you are developing the callus and the plants as we have seen in the picture. So there are methods like 1. Plasmid vector carrier method, gene gun method, chimeric virus method or chimeric virus method, electroporation method. This picture you can see required antigen gene, introduce antigenic gene. By means of the 4 methods is possible. We can select vector system method, gene guns method, or you can say chimeric virus method or electroporation method for introduction of antigenic gene into the plant. For the method of transformation of the DNA or gene into the plant. And then the transgenic plant have been developed with antigenic gene as shown in the picture that can be an edible products of the plant, maybe like tomato or banana or rice or maize or potatoes or carrot or beans or you can see lentils or any edible part of the antigenic gene can be orally consumed or orally delivered to the human health. It becomes an oral immunization. That's called the edible vaccination, edible vaccines. Plasmid vector carrier method means one way of generating edible vaccines depending on the microorganism species to deliver into the plant cell the genetic blueprint for an infectious agent or microorganism antigens, proteins that elicit a target immunological response within the recipient plant. So that can be done by plasmid vector carrier method. Gene gun method, the gene can contain DNA coated metal with gold or tungsten. Particles are fired in the plant cells by means of a gene gun. Chimeric virus method, plant viruses are genetically modified to carry the desired gene and used to infect the natural host such as edible plants where the cloned gene are expressed to varying degrees in different edible parts of the plant. Electroporation method, the introduction of DNA into the cell is done by exposing the cell for a brief period of high voltage electrical pulse. High voltage electrical pulse that is assumed to induce transist pores within the plasmalemma. That means a thin layer of tissue that covers the surface. That place within the plasmalemma, where that is the thin layer of tissue that covers the surface. There you can get the introduction of DNA. That is electroporation method. Plant used for edible vaccines are tobacco, potato, banana, tomato, rice, lettuce, soybean, alfalfa, carrot, peanuts, wheat, corn. This is just to show you the lettuce is also nowadays for salad we are commonly using, and also alfalfa is having many nutrition in the banks of lakes and many roadsides we can see in a watery area. This alfalfa are very common. So just two pictures. Others, all the things may be known very well for the student. Banana are sterile, so the gene do not pass from one banana to another, which is the main reason why bananas are a good choice for edible vaccine. Rice, an edible vaccine using genetically altered rice is used in cholera treatment. Cholera vaccine exists but provides short-lived protection and requires refrigeration. Maize plant generate a protein that is used to develop the hepatitis B virus vaccine. It is cheap and does not need to refrigerate it. A major disadvantage of this vaccine is to be cooked for use, which causes degradation of proteins. Potato, a potato-based vaccine used to combat the Norwalk virus, stomach virus, which is spread by contaminated water and food and cause severe abdominal pain and diarrhea. Potato has been also served as a vehicle for diabetic related proteins, the vaccine against a strain of Escherichia or E. coli, Escherichia coli cholera vaccine. Mason et al., the group of scientists, were the first person who conducted the vaccine-based assay produced in potato Solanum tuberosum to against LTB strain produced by E. coli in maize. Tomato. It has been reported that tomato can be served as a vector to develop the vaccine against anthrax, rabies, and HIV/AIDS. It has merits such as grows quickly, cultivated broadly, heat stable, and high vitamin A composition may boost immune response. Tobacco. Human papilloma virus HPV or the causative agent for cervical cancer and also involved in skin, head and neck tumors. Applications of edible vaccines, malaria. Three antigens are currently being investigated for the development of plant-based malaria vaccine. Merocyte surface protein, MSP, merocyte surface protein 4 and merocyte surface protein 5 for Plasmodium falciparum and merocyte surface protein 4 or 5 for Plasmodium uvuli. Wang et al., the scientist Wang and group of people have demonstrated that oral immunization of mice with recombinant merocyte 4 and merocyte surface protein 4, merocyte surface protein R5 and merocyte surface protein 1 co-administered with cholera toxin B, CTB as a mucosal adjuvant induced antibody response effective against blood stage parasites. Number 2, measles. Mice fed with tobacco expressing MVH measles virus hemagglutinin from Edmostone strain could attain antibody titers 5 times the level considered protective for humans, and they also demonstrated secretory immunoglobulin A in their feces. Carrot, banana and rice are the potential candidate. Candidate means potential edible part which is developed as an edible vaccine. Hepatitis B significantly extended the protective level of 10 milli international unit per ml in humans. Potato-based vaccine against hepatitis B have reported the amount of HBsAg, that means also it is known as HBsAg, means also known as Australian antigen, is a surface antigen of hepatitis B virus HBV. Need for 1 dose could be achieved in a single potato. Stopping autoimmunity, the transgenic potato and tobacco plants when fed to non-obese non-obese diabetic mice showed increased level of immunoglobulin and antibody associated with cytokinins that suppresses harmful immune response. Number 5, cholera. The plant were transformed with the gene coding B unit of the E. coli, heat liable enterotoxin, LTB. The transgenic potatoes expressing LTB were found to induce both serum and secretory antibodies when fed to mice. These antibodies were protective in bacterial toxin assay in vitro. This is first proof of concept for the edible vaccine. So when we come to advantages of edible vaccine, do not require administration by injection, possible production of vaccine with low cost, do not require separation and purification of vaccine from plant materials, necessary of syringe and needles not required, economical in mass production and transportation, heat stable, eliminating the need for refrigeration. Disadvantages of edible vaccine: development of immunotolerance to vaccine peptide or proteins, consistency of dosage from fruit to fruit, plant to plant, and generation to generation is not similar. Stability of vaccine in fruit is not known. Dosage of vaccines would be variable. Selection of best plant is difficult. Contained food like potato are not eaten raw, and cooking the food might weaken the medicine present in it. Not convenient for infants. Safety aspects: contamination through cross-pollination. Vaccine antigen may affect the browsing animals. Vaccine contamination via plant debris spreading on surface and groundwaters. Effects on human living in area drinking vaccine polluted water or breathing vaccine polluted dust. Cultivation and production of pharmaceutical crops should be limited to control the production facilities like greenhouse, or in plant tissue culture that prevents the environmental release of biopharmaceuticals. Now I have added an additional as per the question asked in university examination, 2 mark, and they have asked even 5 mark also. So just I have added, it should be in the initial stage of plant tissue culture. But by means of seeing the question paper, I have seen this is a short question, a small question which is asked like various phases of growth of plant tissue culture they have asked. So the various phases of growth in plant tissue culture, the stages of plant tissue culture which we have seen in the first class itself, first lecture. The section of preparation, that is a 0 point. Initiation of establishment stage, the first one by aseptic manner, preparing explants. Multiplication stage like callus, suspension culture, and next one is rooting, and the 5th one is accumulation, hardening, and becoming as a plant and transferring of the 4th stage. So you can see the formation of growth medium is selected based on the stage of the plant is in or with B entering. But the phases of the growth in the plant tissue culture are as follows, as shown in the graph. Lag phase. Number 2, exponential phase. Number 3, linear phase. Number 4, declaration phase. Number 5, stationary phase. So when we talk about the lag phase, most biological system shows an initial growth phase which is slow. Number 2, exponential phase. In the tissue culture, following a period during which the growth rate of the cells gradually increases, the cells growing at a constant maximum rate, and this period is known as the log or exponential phase, and the increase in biomass concentration will be proportional to the initial biomass concentration. Number 3, linear phase as shown in the picture, a further period of rapid cell division resulting in a linear increase in number, and the phase is called linear phase. Number 4, declaration phase. The declaration in the rate of cell division is due to the gradual reduction in nutrition availability, an increase in waste concentration, and a reduction in amount of free space in culture container. And number 5, stationary phase or plateau phase. Cell proliferation slows down due to the growth limiting factor, such as the depletion of an essential nutrient and or the formation of inhibitory product, resulting in a situation in which growth rate and the death rate are equal. Cells are mostly susceptible to injury at this stage. Thank you for being this class, and please make yourself your own notes and study well, and have a great day.